Despite of high incidence of dengue and Zika outbreak, particularly in the tropical and subtropical regions of the world, there is no effective vaccines or antiviral treatment currently available. Assalamualaikum warahmatullahi wabarakatuh and good day everyone. Hi, my name is Dr. Siti Nur Khadijah Adis. In the previous video, you have learned about treatment and myth about dengue and Zika. Now, in this video, we will further explore on the development of antiviral and vaccine against dengue and Zika virus. In this video, you will learn about the development of different antiviral and vaccine against dengue and Zika that is currently in the pipeline. From that, you will be able to identify the challenges and issues involved in the developmental process. In the end of this video session, you should be able to describe the development and challenges of antiviral against dengue and Zika, and you should be able to describe the development and challenges of vaccine against dengue and Zika. Today, there is no clinically approved antiviral against dengue and Zika. However, a great effort has been made in search for antiviral candidates by using different approaches and methodologies, from testing specific compounds with known antiviral activity to screening of libraries with hundreds of bioactive molecules targeting directly on the virus or on host. Here, in this video, an updated review of the current progress on dengue and Zika antiviral will be presented. In the current search of an ideal anti-dengue and anti-Zika drug, a broad-spectrum activity is particularly desirable to prepare for the next flaviviral epidemic. Potential molecular targets for broad-spectrum anti-flaviviral compounds include viral protein, such as the viral protease or polymerase. Numerous compounds with broad-spectrum anti-flaviviral activity have already been identified by target-specific or phenotypic assays. A viral target protein should ideally combine two attributes. It should be essential for the viral cell cycle, and it should have a low rate of allowed non-lethal but resistance-conferring mutations. In the host target antiviral, there are numerous factors need to be considered during the drug developmental process, as flaviviruses interfere with the host cells in numerous ways. Some cellular pathways may be upregulated to promote replication and entry, including alpha-glucosidase and proteins involved in nucleoside biosynthesis, whereas other functions, particularly those related to the cellular immune response, are suppressed by the virus. Interfering with processes exploited or controlled by the virus has therefore long been considered a conceptually promising route towards antiviral treatment, albeit with limited success so far. After knowing the issues and current development of dengue antiviral, you might be wondering what are the issues and challenges in dengue and Zika antiviral development. Currently, dengue and Zika are considered as neglected diseases as it affects on some geographical region and population in the world. This factor had contributed significant effect on the development process, where the potential of commercialization is rather limited. Symptoms of dengue and Zika are broad, ranging from asymptomatic to severe to death. Thus, variability of the pathological effect has made the development process rather complicated and of less interest when the current simple symptomatic treatment and fluid management is proven effective. Thus, current attention in order to combat the disease is by primarily vector control centered around outbreak area. As far as dengue and Zika vaccine are concerned, to date, there is no clinically approved vaccine for dengue and Zika, but a number are on pipeline on clinical trials. So, what are the goals of dengue and Zika vaccine? The answer is to elicit protective antibodies against the virus as to prevent infection and severe disease, also to minimize the possibility of side effects and adverse reactions. One major challenges faced by scientists in flavivirus research is the unavailability of suitable animal model to mimic and evaluate the real dengue and Zika infection. Dengue vaccine development has been in progress for more than 60 years since the virus was first isolated, and it is now accepted that an ideal dengue vaccine should achieve the following criterions. First, the vaccine should provide a lifelong protection to all 4 serotypes of dengue virus with minimal to zero side effect. And a potential vaccine must be a tetravalent. Second, the cost of vaccine should be lower and affordable for developing countries where dengue and Zika is endemic. And third, immunization of this vaccine should be safe enough and should not increase the risk of severe diseases in individuals with pre-existing immunity. Despite of various challenges in the development of dengue vaccine, there are currently a number of candidate vaccines on clinical trials. And some of them are the live attenuated tetravalent vaccine, namely the CYD-TDV developed by Sanofi Pasteur, the TV003, TV005 developed by the National Institute of Health, United States of America, the chimeric dengue vaccine based on dengue virus 2 developed by Takeda. Other than that, there are also purified inactivated tetravalent dengue vaccine which now evaluates its effectiveness in different adjuvants. And finally, subunit protein vaccine comprised of truncated envelope protein from each of the 4 dengue serotypes. Meanwhile, in Zika vaccine pipeline, there are currently a candidate of a DNA vaccine, which is a DNA-based vaccine comprised of Zika genome, purified inactivated vaccine, live attenuated vaccine, and also mRNA vaccine. Great effort is currently being put to produce this vaccine with high efficiency and relatively low side effects. It is hypothesized that traditional use of mud crab soup helped to relieve symptoms of dengue virus. Nonetheless, the antiviral property of mud crab extracts against dengue infection have never been experimentally addressed. In the search of effective antiviral for dengue, scientists here at UMT are now focusing on the fundamental analysis of mud crab antimicrobial protein as a potential dengue virus inhibitor. The study involved in vitro-based analysis of protein expression and testing in cell-based antiviral assays. This study, which focused on a medically important flavivirus, is timely as the only treatment available of dengue patients is supportive care due to the unavailability of clinically approved antiviral therapy. Now, having watched this video, I hope you can now explain on the current issues and challenges in dengue and Zika antiviral as well as vaccine development. As previously explained in this video, can you identify the advantages and disadvantages of each type of dengue and Zika vaccine strategies? Thank you.